Oh, hi everyone. So welcome to BioTuring Webinar Series, AI in Biology. So today is my great pleasure introducing Dr. Bo Wang, and today he will present scGPT towards building a foundation model for single-cell multiomics using generative AI. And you know, for the last year, we have seen a large wave of AI, especially with ChatGPT. And the field of AI is moving so fast right now. And even when I see the paper scGPT in that Bo Wang put in bioRxiv, I got so many requests from people, colleagues, asking to invite Dr. Bo Wang to present his work. So even though the paper is not yet in peer-reviewed yet, but I have to note that some of my professors got a field medal for just putting his papers on archive. So Dr. Bo Wang is a tenure-track assistant professor in the Department of Computer Science and Laboratory Medicines and Bathobiology at the University of Toronto. And he's the inaugural Temati Professor in AI Research and Education in the largest research hospital in Canada. He also holds a CIFAR AI chair at Vector Institute, and CIFAR stands for the Canadian Institute for Advanced Research. Dr. Wang obtained his PhD in computer science at Stanford University, and he has been focusing on machine learning, computational biology, computer visions, with a particular emphasis on their applications in biomedicine. And his contributions in this field has led to the recognitions through numerous esteemed awards, including the Gärtner Early Career Research Award and also the Canada Research Chair Award. And, today I and many people in the audience here, looking forward to hearing Dr. Bo Wang's talk in scGPT and how to use the foundation. What is it? How to use the foundation model for single cell analysis. All right. So that said, the stage is yours, and from time to time, there can be some questions, and I may interrupt you a little bit, just a little bit, to ask questions. And then, there will be also questions at the end of the sections as well. Okay?
Sounds great. Sounds great.
All right. Thank you so much.
All right. Hello, everyone. Let me share slides. So let me know if you can see the full screen.
Yes,
Awesome.
you can see this full screen.
Awesome. Great. Okay. Hello, everyone. Thank you for inviting me to present our work. So this work is a recent research paper from our lab focusing on building foundation model in single cell. So before we start, just want to make some claim that this is still ongoing work. It's under review. And we put the preprints and also shared all the code and weights data in this GitHub link. So feel free to check it out. And we definitely welcome your comments and feedback. All right. So the talk is about foundation models for single cell. But the first question people often ask is, what are foundation models? So foundation models, in a more academic sense, was first coined by a seminal paper from a group of researchers in Stanford, which I share on this slide. You can see lots of authors on this paper. And really the core idea of foundation model is that it is a system that can absorb common knowledges from large amount of data, such as text, such as images. And then once the model is trained, we can adapt this foundation model to many downstream tasks with none or very few data for each specific task. So this is a core idea behind foundation models. However, that paper has 300 pages, too long, don't read. I summarize some of the key features to build a good foundation model. So in my summary, I summarize as A, B, C, D. So here, A, algorithms. Most of foundation model are based on transformer-based deep learning architect, which I will briefly talk about these architect later on. B, business or applications. So basically the idea of foundation model is that one foundation model can enable multiple tasks by transfer learning. And C stands for computing. Most of foundation model needs specialized hardware, such as GPUs or TPUs if you are from Google. And last, D, data. Building foundation model requires massive amount of data for pre-training, so which we're going to talk about for scGPT as well. All right, so all of these recent amazing achievements of generating models such as ChatGPT in recent years really motivate us to think about the question, can we build a foundation model for single cell data? Since I have been researching on single cell data even from my PhD time in Stanford. And the motivation is quite clear. While texts are made up words, cells can be characterized by genes. So this analogy inspires us to explore foundation model for single cell. And although here I want to be sure that we are not claiming that the internal mechanism of biology and language are the same, but there's enough technical similarities really to motivate us to explore this direction. And in the long run, we expect such foundation model has the potential to gain general understanding of underlying biology, just as foundation models such as ChatGPT can have a general understanding of languages. But we are not sure about this is a good idea. So we want to check whether we have all the key features A, B, C, D as I outlined before. So let's look at this A, B, C, D one by one. So A, algorithms. As I mentioned, most of the foundation model we see today are based on this seminal architecture called transformers. This was published in 2018 from Google, titled "Attention is All You Need." And if you look at this transformer architect, it has two parts. Left is encoders and right are decoders. So you have encoder to encode the text into a latent space, and then you have another decoder to generate the corresponding output. And this, since its publication, this transformer architecture has been widely used and it paves the foundation for many large language models we see today, such as ChatGPT. And if you stack enough encoders, and you really get BERT type of model, which was, again, originally invented by Google. And we also see similar models in single cell recently, such as scBERT or scFormer, which is really our first attempt previously in using transformer in single cell data. And also recently, Nature also published GeneFormer. So the similarity between all these models is that they all use encoder-like model to basically project single cell data into a latent space. On the other hand, if you stack enough decoders, you get GPT-like models, which was first invented by OpenAI. And the key difference between GPT-like model and BERT-like model is that GPTs are generative, and therefore it has better capacity in modeling the true data distribution, and has many so-called emerging properties in the sense that it can do very well on zero-shot or few-shot learning. So this is also reason why ChatGPT has such huge success lately. And build a similar model like GPT, our scGPT use GPT-like model to learn how do we generate single-cell data. So we have the algorithms. Now let's look at B business or applications. As everyone knows, there's no shortage of tasks in single-cell domains. I will just list a few common tasks here, such as cell type clustering, batch integration, multi-omic integration, gene perturbation prediction, gene regulatory analysis. These are common applications we explored in our scGPT paper as well. Okay, let's move to C, computing. Not much to say here. We were very lucky to have some resource from Vector Institute, which is the AI hub at Canada. And also we have some support from Computer Canada, but still quite limited. I'm sure this is a challenge often met by individual academic lab nowadays. Last, D, data. Everyone says data is a new oil for AI. And same for scGPT, we do need a large amount of data to have a good training for scGPT. And thanks to initiatives such as Human Cell Atlas, we now see a surge of single-cell data that are made public to all the researchers in this area. And particularly, I want to kind of shout out to cell by gene census. It really helped our development of scGPT. And we continue to work with this data set to further develop many tissue-specific foundation model recently. All right, so with all that said, we seem to have all the 4 components ready, and we are confident that we can start to build scGPT. That was exactly what we did for the past few months. So here is an overview of scGPT. scGPT consists of 2 steps. First, generative pre-training, and second, fine-tuning for downstream tasks. So let's look at these 2 steps in more details. We started with, originally we started with blood cells, and then once Cell by Gene published their 33 million cell atlas, we immediately switched to Cell by Gene collections. So for pre-training, we collected 33 million human normal cells from CZI Cell by Gene census. And as you can see from this figure, these 33 million cells cover quite a few major organs, such as brain, such as blood, such as lung, heart, and overall we have 33 million human normal cells. So note that these cells are all healthy cells. And then once we collect these 33 million human normal cells, we pre-train our scGPT. And once the scGPT is trained, we randomly sample about 30,000 cells and draw a UMAP, and just to have a kind of sanity check to see whether it learned anything meaningful. And we actually find quite nice hierarchy of cell types. This gives us confidence that this model is well trained. And we're going to use the next few slides to briefly talk about some of the technical details we used in pre-training. All right, how do we do the pre-training? First, we have to define our input. The input to scGPT contains 3 sets of tokens. First, gene identifiers, basically just gene names. We project these gene names to tokens, just as ChatGPT does, to project words into tokens. And the second input is expression values, because for many single-cell RNA-seq data, it's not binary values, although we can do binarization, but we want to keep the original expression values. So what we did is we binned all the expression values and then project this binning into tokens. The third input to scGPT, we call it condition tokens. So it is a mechanism we designed to incorporate a lot of metadata. For example, we can use these condition tokens to indicate which organ this cell comes from, to indicate which studies, which technology this comes from, or for perturbed data sets, we can denote which genes are perturbed. So this design of condition token really give us flexibility to incorporate many metadata for us during the pre-training. Once the input is set, really the core of scGPT, as I mentioned previously, consists of stacks of masked multi-head attentions. And in the current version, we have 12 blocks, and each block has 8 attention heads. So relatively, it's a small model in comparison to bulky foundation models such as ChatGPT. But still, we think it's enough to digest these 33 million cells. As we have more and more cells, we may need to increase the capacity. But for the current version, we stopped at about 100 million parameters, which consists of about 12 blocks of attentions. And then once we have the input, we basically just input these input tokens into blocks of transformers. And really the core of the core is this design of the masked multi-head attention module. It is not a direct plug and play from NLP community, since single cell is different than languages, in that words in a sentence are sequential, while genes in a cell are non-sequential, right? For example, a word says, I like coffee, is different than the sentence coffee likes I, right? So the order in a sentence actually matters for languages. However, it's not that important in single cell RNA-seq. In order to make the adaptations, the innovation we made here is following. So the generative process consists of 3 steps. We start with a starting token, CLS, which we treat it as a cell embedding for each cell. And during the attention phase, we rank the genes based on certain orders. So in our scGPT, we order the genes based on its attention value to the cell embeddings. You can have other orders, such as predefined order of pathways or even chromosomes. But we find that simply order the genes based on the attention values works out very well, because attention values in other aspects really indicates the importance of genes to this specific cell. Now, once we have the ranked genes, we segment the whole gene list into a few blocks, with each block consisting of a percentage of all the possible genes. And then we basically use autoregressive masking to mask the later blocks of genes and predict these masked genes using previous predicted genes. So very similar to how ChatGPT pre-trained itself. And then once we kind of rank the genes in a specific order, we can pre-train scGPT using autoregressive manners. And I also want to mention a very important trick we used during pre-training. We call it teacher forcing. This is also a trick widely used in training language models such as ChatGPT. The basic idea of teacher forcing is that during the early epochs of training, because your model is initiated randomly, so the beginning epochs, the model does not understand anything. So it's better to use the original gene expression values instead of using the predicted gene expression values. And this trick has been shown very important in the early epochs. But after a good jumpstart, we can use the traditional autoregressive training. All right. So once the pre-training is finished, we can take this base model and perform fine-tuning on some of the downstream tasks. And the fine-tuning also contains lots of innovations in terms of the design of loss functions, particularly when we have multiple batches. The loss function consists of 2 types of objectives. Specifically, we have a self-supervised objective in which we want to force all the neighboring cells to have the similar embeddings from scGPT. So this is commonly used loss function for batch for a single cell. The other loss function we find very useful for multi-batch integration is called adversary loss. Basically, the idea is very simple. We want to construct a classifier that cannot distinguish which batch this cell comes from. In other words, we can remove the batch effect. And with these loss functions, we can do a very good job in fine-tuning to integrate multiple batches And the way we do fine-tuning is quite straightforward. We basically use a standard pipeline in deep learning. You design the loss function, you do SGD to do back propagation, and then you update, you kind of update the weights in a fine-tuning way. So it's a very standard pipeline in deep learning. All right, so we then once we have the model, we want to test how good our model is. So we collected PBMC 10K dataset, which consists of multiple batches, and this result we compare with some of the state-of-art specialist approaches such as Seurat v3, SCVI, and Harmony. These are widely used algorithms for batch integration, and we compare scGPT by reporting the average bio. Average bio is basically an average value of how good this algorithm can remove batch effect and how good this algorithm can preserve the cell types. And you will find that scGPT, just through fine-tuning, does a better job than Seurat v3, SCVI, and Harmony. These are specialized approaches. And similar workflow can also apply to multi-omic integrations. So here we really only scratch the surface of multi-omic integration by only integrate single-cell RNA-seq and single-cell ATAC-seq by using another 10x multi-ohm PBMC dataset. And similarly, we compare scGPT with Seurat v4 and scGLUE, and these are widely used multi-omic integration algorithms in current practice. And again, we compare with average bio metric. We find that scGPT does a better job in comparison to other specialist approach. We also tested in the setting of mosaic integration where not necessarily paired data are given, right? So mosaic integration represents a very challenging task where you don't have paired RNA-seq and ATAC-seq. Instead, you have unpaired different modality omics, such as RNA-seq, ATAC-seq, even proteomic, but these are unpaired, but they come from the same population. And we compare with another specialist approach, which is called scMOMAT, a recently published paper that achieved state-of-art in mosaic integration. And we compare with scMOMAT, we find that scGPT does a better job in not only removing the batch effect that are coming from different modalities, but also did a great job in preserving the cell types. So we are confident that scGPT with proper fine-tuning can do multi-batch integration, multi-omic integrations. And then we move ahead. We want to test how good scGPT is for cell type annotations. Cell type annotation is a widely used tasks in single cell community. And the idea is to assign cell types to some unseen cells. So in order to have this test, we collect the 3 data sets, and even including a cancer data set, pancreas cancer data set, also some other immune cell cell type assignment data set. And we report 4 metrics: accuracy, precision, recall, and F1 values. And again, we compare with 2 other methods, a state-of-art method in cell type assignment, scBIRD and Tosica. And we find that scGPT does a much better job in reporting accuracy, precision, recall, and F1 values. However, this is a supervised task in which we have a reference data set. We also have a holdout test set. So people may wonder, how good is your zero-shot capacity? So basically without any fine-tuning. So we did the test on the COVID-19 data set by zero-shot scGPT, and we find that even zero-shot without fine-tuning, scGPT actually can capture very well on the cell types informations. So we on this COVID-19 data set with more than 20 cell types, scGPT can achieve high accuracy of almost 86% accuracy on query set. Query set is a set we never see before, by just simply propagating the reference labels. So with this experiment, we want to extend further. Can we do reference mapping from large cell atlas, right? We have access to large cell atlas nowadays. You probably see lots of Nature paper publishing every day to publish atlas these days, right? So we want to extend scGPT to reference mapping where we don't have any supervisions, we simply run zero shot. So we designed this pipeline into 3 steps. So first, we use foundation models such as scGPT to generate cell embeddings on that cell atlas. And then we try and store the search index using a package called FAST, which is a super fast and efficient pipeline for indexing. And then once a new data set comes in, we use nearest search and propagate the labels to this new data set. So very simple 3 steps. And then we test it on cell by gene census, which has 33 million cells. And we find that first, the index we saved is actually a very low weight. It only occupy less than 1 gigabyte. So this is memory efficient. Second of all, it's time efficient. So the search, let's say you have a new data set with 4,000 cells. In order to search the whole reference of 33 million cell atlas, it only costs 7 seconds on CPUs. And if you are lucky to have a good GPU, it only takes less than really 1 second. So the reference mapping is extremely efficient, but also accurate. So now we are actually collaborating with Cell by Gene to provide this functionality to users who use Cell by Gene, in the sense that whenever they generate a new single cell data, they can simply run scGPT and query the nearest neighbor cells and then propagating the cell types from cell atlas. So moving from cell type annotations, we actually find a very interesting phenomena for foundation model. It's called scaling law of foundation models. So the experiment is as follows. So we want to understand the effect of training data size to the performance of these foundation models during pre-training. So what we did is we randomly sample 30,000 cells or 300,000 cells, and then 3 million cells, and then 33 million cells. 33 million cells means the whole data set, all right? And then we use these subsampled cells to pre-train scGPT and then report the accuracy on a holdout test set. And we find that the accuracy increases a lot when you increase the training sample size during pre-training. This is observation 1. So this is very easy to understand. That's why we need large amount of data sets. This is why also ChatGPT, for example, train almost all the text you can find on internet, because the more data you train, the better the model becomes. Another observation we also find is interesting, that once you have 3 million cells during pre-training, the performance gain starts to diminish. Although it still increased a little bit, but the performance gain actually diminished quite a lot. So this is a very interesting observation. Makes us wonder, is that how many cells are good enough, and what are the criteria to choose cells for your pre-training? But this is a good, very interesting observation we found and in training scGPT. And also then we move ahead to test other tasks such as gene perturbation response. Again, we did the same practice we did before. We choose quite a few benchmarks that are widely used in gene perturbation prediction tasks. And then we compare with some specialist approach such as GEARS, which were recently published in Nature Biotech. And these are considered state-of-the-art approach on this specific task. And we find that same model, simply just fine-tuning, scGPT can beat the specialist approach on gene perturbation predictions. And similarly, not only scGPT outperform on tasks related to cells, we also show good capacity on tasks related to genes, because this is really a good side product of these foundation models, such as scGPT, in that not only we learn good embeddings of cells, we also learn good embeddings of genes. Because remember, during training, we map the genes into tokens. We learn these gene embeddings during the attention modules. And what we test is gene-gene interaction analysis. And by simply zero shot on some immune cells, we find some really good gene families or gene modules that had been discovered before, such as HLA families. We also compare with traditional pathway enrichment analysis from Reactome by simply calculating the co-expression network. And we find that by calculating the gene regulator network from scGPT, it can find a lot more differentially enriched pathways than the traditional co-expression networks. So this gives us the confidence that scGPT can learn very good gene programs during pre-training. And another fun experiment we find that is because cell by gene has lots of atlas data for certain specific organs, so we want to test organ-specific foundation models. So what we did is we only train scGPT on certain set of cells from one single organ. For example, we train a foundation model for brain. We train another model for heart. We train another model for blood. Similarly, we train other foundation model for single organs. And we want to see which foundation model serves the best starting point when you have a new data set. And particularly, we took a COVID-19 data set because most of the pre-training cells are healthy cells. And we want to see when you have a disease cells, what's the best starting point for your foundation model. And the observations are very interesting. There are twofold observations. One is that if you start with organs that are very similar to your disease, for example, for this COVID, we find that if you're starting with blood, if you're starting with lungs, we call it in-context models, the results are much better than if you start with out-of-context models, such as kidney, brain, or pancreas. We also find another observation, which is interesting, is that it always serves as a good default value if you start with the whole body, which are trained with 33 millions. And the intuition is that these whole body models has seen 33 million cells. It has covered lots of heterogeneity. So therefore, no matter what data you have, it's always a good starting point for you to start fine-tuning. And since its publication, we got lots of attention through the community and lots of requests saying, can we use your model? But we want to use your model, but we don't have the GPUs. Can you help us? So in order to make sure these foundation models are accessible to everyone, we created a website called scGPT Hub. And basically, this is a collaboration through Amazon and AWS Cloud. And the idea, maybe I can just play the video. Yes, I can play the video. The idea is that we build this web portal. And for users, you basically just sign up. And then you upload your own data set. And then you choose your default model, foundation model to start with. You set some hyperparameters you want. And then you just submit your job. And usually if you have 10,000 cells, it takes about 3 to 5 minutes for this job to finish. So it's perfect time for you to grab a coffee. And then once you finish your coffee, you come back and all the results are in your emails, and you just download all the visualizations, all the embeddings from scGPT. You don't need to have your own GPUs. And you will also find that the fine-tuning is actually very lightweight. It only costs one normal GPUs with 3 to 5 minutes for a data set of 10,000 cells. So it's very efficient to use this web portal. And since its publication, I think it's 2 months ago, we already have thousands of users on this scGPT Hub. So again, I want to say, if you are interested in using scGPT but don't have GPUs of your own, feel free to sign up. Feel free to test out this scGPT Hub. All right, so that basically summarizes most of the technical details and applications of scGPTs. However, I do want to emphasize some of the existing limitations because this model is really one of the very first attempts to build foundation models in single cells. Some of the major limitations we see from scGPT are the following. So first, scGPT is mostly limited to RNA-seq. Although in our paper, we discussed how we can use scGPT to integrate other modalities such as ATAC-seq, but we really only have a well-trained base model, foundation model for single-cell RNA-seq because of data limitations. Cell by gene data consensus, they simply don't have a large data set of ATAC-seq that are well curated. So we basically limit ourselves on RNA-seq. However, again, as I mentioned, the bottleneck is really data access. Once there's a resource with large amount of ATAC-seq, we can easily build another foundation model for ATAC-seq. A second limitation is that scGPT, despite of learning meaningful gene programs, is still limited in causal inference, meaning it's still guided by associations. We don't think scGPT can learn causal mechanisms of single cell data yet. Although it's a very interesting research topic, there's some ongoing research in our lab to see how we can better interpret scGPT, how we can better learn some causal relationships. But so far, the current version of scGPT is still guided by associations. The last limitation we see from scGPT is that the pre-training is quite compute intensive. For example, in our scGPT, we train on 33 million cells. We have about 100 million parameters. It took us 12 A100 GPUs for almost 2 weeks. So that's very compute intensive and cost lots of GPU hours and also cost many times to really do a good job in pre-training. However, the good news is that we did all the hard work for you so that we share the model so that you have access to the foundation model. What the user needs to do is simply fine-tuning, which is very lightweighted, as we show on some of the experiments. So how do we move forward with foundation models in single cell? What I hope to see where scGPT can lead the change is following. So in current practice, most of the single cell research are hypothesis-driven approach rely on individual biologists' prior expertise. What we hope scGPT can lead is a data-driven approach that can be flexible enough to incorporate expertise priors, but mostly driven by large amount of data. Current practice in single cell often involves small data set, single center validation. With scGPT, we hope we can move to large scale data validation to multi-center validations by connecting us to Cell Atlas. And also in current practice, many single cell bioinformatic pipelines are designed in a stepwise way. Every step, such as batch integration, cell type assignment, gene perturbation prediction, every step requires the output of previous steps, and different steps use different state-of-art methods, and often these methods have very different assumptions of data distributions. So some of the assumptions may conflict with each other, right? So what we hope scGPT can lead is a general use framework that one model solves multiple downstream tasks all in once, and in the end-to-end approach with optimized frameworks such as GPUs, so that it's more efficient. However, in order to achieve these 3 goals, we see collaboration is essential because we need to have a bridge to connect with computer scientists like myself to many biologists or clinicians who have a much in-depth understanding of the wet lab data sets. So how do we collaborate is actually very important to have successful interpretation of these foundation models. Despite its high popularity of foundation model in single cell community, we do want to say that we should move forward with care, because this concept is quite new and there are a lot of unsolved problems with foundation models. And I list 4 major issues, but there are a lot more. Particularly, it's not very clear to us what are the best practice to curate this large-scale pre-training data beyond a single-cell RNA-seq. How do we curate, for example, spatial omics? How do we curate proteomic or taxi? There's no standard kind of recommendations yet. And second of all, I think it's one of the major issues is we actually don't know how to evaluate various foundation models in an unbiased way. Since our scGPT, there's been some other foundation models popping out very often, and everyone claims it's a foundation model, but we actually don't know how to best evaluate these different foundation models. And also, third of all, how do we make sure foundation models are open and transparent? Because most of foundation models are very bulky, and sometimes it's not clear to us how do we understand these foundation models. And last but not least, how do we make sure to make sure these foundation models is accessible to everyone? So the attempt we make is scGPT Hub, but I think that there should be a more more direct way to make sure these foundation models are shared across the community. All right, with that, I want to acknowledge really the key contributors, my 2 students, Houghton and Chloe, and also want to thank Connor and Gary for the help with building scGPT Hub, and also thanks to all the lab members. And I want to say, if you are interested in using scGPT, please don't hesitate to reach out. I'm more than happy to discuss potential collaborations. And with that, I'm more than happy to take any questions.
Thanks so much for this excellent presentation. So started with questions, okay? So very easy questions from me. So how to avoid, what is hallucination in this context, and
Yeah. So hallucination is an issue it's really the holy grail of natural language processing nowadays. And we see hallucination from ChatGPT very often, particularly when it comes to references, right? It made up reference when it cannot find the right one. However, it's not clear to me how hallucination works in single cells. On the other hand, actually, as Aviv, which is the leader of single human cell atlas, she pointed out that she's actually interested to know how these models hallucinate, because hallucination in biology may be beneficial to generate new hypotheses, right? So we had a lot of internal discussions about how do we distinguish between hallucination and errors. And we also had lots of discussion about how do we distinguish between hallucination with correct but unseen hypothesis. It's not very clear to us yet, and this is definitely something very interesting to me as a research topic. This again calls for better interpretability or causal understanding of these foundation models.
Thanks so much. The second question, how would the retrain model work if we have, let's say, not 30 million, but 200 million cells?
So the pre-training, as I mentioned, is a task with heavy resource. We also share some of the scripts to do pre-training. The idea is generative pre-training. You kind of mask certain amount of genes, you use the remaining genes to predict the masked genes. So the algorithms and all the training scripts are shared. However, I do want to point out that in order to have a decent pre-training model, very often you need to know many engineering tricks, just as many other deep learning models, right? You need to understand how to set the learning rate. You need to understand how to set the batch size. How do you do parallel computing between different GPUs, things like those. However, I think we share lots of recommendation and experience in our transcript, in our scripts, and hopefully these can help with training larger foundation model given new data set. We're actually collaborating with other Atlas providers to see can we train a foundation model for them, you know.
Okay, so the next question, going to a little bit more technical, is would you extend the merits of GPT over BERT architectures, and why did you choose GPT?
Yeah, very good question. So we actually started with exploring BERT like model. We actually published SC Farmer, although we didn't train on 33 million cells. And then we further train scGPT because we believe that generative modeling is more powerful than simple representation learning. Like BERT is an encoder that basically learns the representation only. You know, it does not really learn to generate cells, therefore does not actually model the data distribution. This is really the generative pre-training is so powerful. This is again correlating to languages. You will find that ChatGPT is much more powerful than BERT from Google in terms of generating some novel response, right? Because it's generative. That's the power of generative AI.
Thanks so much, Paul. The next question, so how does the model perform on unseen data? For example, in the cell type prediction case, in one data set contains a rare cell type that's not including even in the foundation model training
Right.
data. Would it say that it is an unknown, or would it say that it is
Yeah.
something
So,
else?
right. This is an excellent question because this question is related to generalizations of these foundation models, right? So what we find that we tested on many unseen data, it works quite well, even including zero shot. However, the challenging task is the unseen cell types, as this question really alluded to. We haven't had thorough evaluation that is specific to unseen cell types. However, through some prototyping and experimentation with cell by gene sensors, we actually find that scGPT can, by just simple zero shot, can map these novel cell types into a separate class. And we can actually have a thresholding mechanism says, if you are not confident enough, just say, I don't know, you know. So, or just say, this is a new cell types, which I don't have enough information about from the reference data set. So I believe there's a way to describe this cell type, these novel cell types, through some novel thresholding approaches. And but we definitely need a more thorough evaluation. And also a step further is if you identify this as a unknown, unseen cell types, how do you characterize it? Right. So this requires some interactions with biologists or some more interpretability says, can you find some genes that are defining these cell types? So, and this is again where scGPT, I believe, can provide some benefits because we do learn a very good gene regulatory networks. So maybe by mapping the cells, particularly looking at these gene programs that define these novel cell types, it can provide some hypotheses.
Right, right. And in the threshold that you were mentioning, probably I suspect it can be in some distance in the nearest neighbor from face, right?
Right. It can be the distance, it can be the confidence, so there are many ways, or it can be, both. There are many ways to define this thresholding approach.
Okay. So there's another question from our tool. Zalata, have you tried not binning the gene expression, instead projecting on the continuous value?
Yeah, excellent question. Excellent question. So this is a very technical question. Um, we tried bunch of approach to model these expression values, and the binning is the simplest yet effective way we found, and this is why we chose binning. We also try, as you mentioned, like a setting different spectrum, but then how to set like a disentangled tokenization for these expression values is actually very challenging if you set it as a continuous spectrum, because you
Yeah.
want to see some distinguish between the values. And there's other papers that using kind of binning on depths, the sequence depths to incorporate into that model. I think that's very valuable as well. But this is why, as I mentioned, this is the first attempt to build this foundation model for single cell. There's certainly a lot of ways to improve. This is why we need lots of discussion from the community. But I think the binning is simple yet effective way to model the expression values.
Right, right. Um, so the next question is, how do you standardize the metadata of the input data in the training? And are you using the standardized metadata from the Human Cell Atlas,
Right.
which can
So
be
yeah. Yeah, very good question. So data curation is very important for building foundation models. And to be honest, when we first published the first version of scGPT, we didn't use cell atlas because they didn't publish yet, and we were collecting these 10 million blood cells from our own from literature. It was super noisy. The model was not as powerful as scGPT right now, and then actually it's interesting. On the same day we published the first version, and then Cell by Gene published their 33 million cells, and really without even second of hesitation, we moved to cell by gene and retrain scGPT using the 33 million cells with the standardized curation metadata, as you mentioned, and we find that it works much better.
And
So
also the question, Paul, is
yeah,
that the for the metadata standardizations, how important is it to have multiple type of metadata, like not besides cell type, we also have condition, treatments,
yeah,
uh,
yeah. So,
even
so a
technologies.
very good question. So this is why we introduced the condition token, on top of the expression values. In that we want to use these condition tokens to indicate different metadata, and this is why this is so flexible and can do so much better job in terms of transfer learning, because it's very flexible. Let's compare SCB with SCVI. Let's say you want to transfer learning on SCVI, which is possible. However, you have to do a very strict alignment. You need to say which genes are aligned. It have to exactly match gene alignments, right? For us, we don't need that because we train using all the gene tokens. And then when a new data set comes in with a lot less genes, you can still do that. You basically just take the subset of gene tokens and map the token space on these subset of genes, right? Or same thing for perturbations. You just map the perturbed gene tokens to scGPT and do a little bit of fine tuning. Sometimes even without fine tuning, it works very well. So we believe that this foundation model is a step forward in terms of better capacity for transfer learning.
Yes. How does scGPT compare to GeneFormer?
Very good question. uh, well, we were first to release the preprint, and then GeneForm published in Nature months later.
That's what
Uh,
I know as well.
yeah, so GeneFormer is an encoder type of model. And in our latest version, reviewers actually ask us to compare with GeneForm, and we did that. And we see, particularly on a few tasks such as perturbation prediction, such as cell type assignment, scGPT does a better job in terms of GeneFormers, but GeneFormer is smaller. So in that sense, GeneForm is actually a very good model. The model is a lot lighter than scGPT, but we believe in terms of capacity, particularly on multiple tasks, we find that scGPT is more powerful. And there's actually a recent evaluation paper that compare with all the foundation models, and scGPT is really the only model that have all the box checked. Right? Is it the best model ever? No, I don't think so. But it strike a balance between the accuracy and generalizations and also model complexity.
Uh, I want to select because there are so many questions here. Uh, so you have any foundation models trained as disease specific?
Yeah, so we train so far as we train on healthy data because we see similar motivation from human cell atlas because we think healthy data serve as a good reference, right? So, however, we are very interested in collaborating with different collaborators to see how we can build disease specific, like for example, immune disease or let's say cancers, right? And we're working with some collaborations through Harvard, Stanford, and it's not conclusive yet because we haven't really published any papers yet, but see some preliminary success. But I want to see that we definitely seek more applications in more disease analysis because that's a goal, right? That's why we build this model because it's not just analyze healthy cells. We want to analyze disease. And we do think this is a good first step because once you have a disease model, you can just do fine tuning. And we find that fine tuning works extremely efficient to jump from healthy to disease. I think even we see some users that do fine tuning from mouse to human. We haven't done that yet. From human to mouse. But from some feedback through GitHub, people seems to be satisfactory about the result. So we are very optimistic. This again, comes back to the one of the reasons we are interested in the foundation model is it's much better generalizations.
Also, in another context of having more data better? Let's assume that it is not 30 millions, but another one give you a data set like 10 times as large, like 300 million. Would it be
Correct.
better?
So the answer is yes. That's also the scaling law shows. That's also true for NLP. However, I think different from NLP is that we care more about diversities, heterogeneities of training data in single cell than languages, right? And so far, I think it does not add too much value if we simply adding healthy data from these major organs. Like we already trained on 3 million of blood, another 3 million on brain. So does not add much if you add another 3 million on brain again. However, it will add a lot if you add another 1 million cells of some unseen organs. So far, we trained on 7 organs. So if we add more healthy data from another organs, I think it will help a lot. Also, I want to expand on disease data, you know. So I do believe by continue training this scGPT on disease data across different disease type, it will help a lot. So diversity, the heterogeneity, matter a lot than just sheer sample size.
Yes. All right. So we have like 64 questions left to answer, and the time is actually already coming to the end. So again, Bo, thank you so much for having a great talk. I enjoyed so much, and I believe our audience also, enjoy the really important work here. And we will send you the list of 64 questions left, so that you can, when you have time, you can answer and we can spread
Absolutely.
to the colleagues around. Again, thank you so much for spending time with us.
So
And
thank you for having me. And I just want to say it again. If you're interested in collaborating or interested in exploring scGPT, feel free to reach out. We are a big believer of open science. That's also what we did by sharing all the data weights of scGPT. So please feel free to reach out if you have any questions. Thank you very much.
all right, thank you so much, everyone. Have a good day. Bye bye.